In this study, we sought to understand the mechanism by which distant imprinted genes are silenced by non-coding RNAs. The model system that we used was silencing of the IGF-2 receptor cluster by Airn long non-coding RNA. Airn is known to silence IGF-2 receptor by transcriptional interference with its promoter. So the question is, how are distant imprinted genes in this cluster silenced? The model we came up with is the so-called enhancer interference model, whereby expression of Airn disrupts an essential enhancer for an imprinted gene like SLC22A3, causing it to be silenced. To test the predictions of this model, we first performed chromosome conformation capture, comparing the A3 promoter with the Airn gene, and found an enrichment of maternal interactions consistent with the model. When we truncated Airn, we found that these interactions were also gained on the paternal allele, indicating that these interactions are blocked by Airn expression. To test the model, we then performed a deletion of the entire Air gene in the mouse. We crossed this mouse to a castaneous mouse and performed RNA sequencing to look at imprinted expression over SNPs. And contrary to our model, we found no change in imprinted expression in the maternal deletion for A3 or other imprinted genes in the cluster. In contrast, on the paternal allele, imprinted expression was lost for A3 and other genes in the cluster, consistent with other Air mutants. Therefore, we came up with a revised model whereby these interactions are initially present on both alleles and allow Air to find its targets and to initiate silencing. And then once the silencing is established and you have repressive chromatin, these interactions are lost.